Hello, everybody. This is Owen O'Leary, the Director of Education at the Tuition Centre. I'd just like to welcome you here this afternoon at this point. And you're here to hear, of course, Dr. Declan Cathcart, who's going to be doing a free biology lesson today on genetics, the study of inheritance. And I'm just saying hello to you right now, just so that you know that this is going to start in 5 minutes' time. And we're just going to we just started a little bit early there, just to give everyone the opportunity to log in and to download the notes, etc. You can see there's a link for them there and the Google Drive just in the comment section. Okay, I'm getting the green light here from the tech team here. I think that everything is working. Everything is working. It sounds good, looks good. So I see a comment coming through from one of my students back there in Bandon. So hello to you all, and you're very welcome. Just while we're waiting, so we're just going to wait till 2 o'clock, of course, naturally, for everyone, and just maybe a minute past 2 weeks, naturally, people will be logging on, yourself, then just be tuning in at the time. I'll just actually just to kind of give you a little bit of a rundown of what's going to happen today. So obviously, and a lot of you were here last week for Declan's excellent lesson on which was also on biology and genetics. But just a couple of questions that were asked of me just in the last 24 hours in regards to the Tuition Centre. I'd just like to say well done to all of you who did the chemistry free pre, and well done to you. You got the marking scheme today, so I know a number of you have been correcting that this morning, so well done. Yes, we have the business free pre is there. The link is there on the website now for that. So there's going to be the free pre in business, and that is going to be on Thursday, the 11th of June. The day it was due to be on in the original Leaving Cert timetable. Any of you there who have done the maths pre and have done pre-paper 2 and you have your result, but you haven't inputted yet, we'd love you to input that today, because today is going to be the final day where the results can be inputted. And we're all here to talk about predicted grades, etc. at the moment. And I suppose, look, it is nice for you to have at this grade that you can kind of benchmark yourself off at this time of the year, where you're saying, okay, I got whatever you got, a H6, H5, H4, whatever grade you got today. At least it's a benchmark then that you can kind of think, okay, that's where I was in the month of April, start of May. And so it gives you a guide in your own mind anyway. There's a number of free lessons like today's lessons have been added, and there's going to be a lot more of those added during the week. So just keep an eye on the website for all that. We'll have a number of other free resources. So this lesson here is going to be recorded and is going to be on our YouTube channel. So if you check out our YouTube channel, you will obviously see there's other resources there. And in the next days and weeks, there's going to be a lot more material added to that. Let me just talk about today's lesson. I'll introduce Declan formally now in a moment, but just for those of you just tuning in, this is Owen O'Leary here, the Director of Education at the Tuition Centre. And we're just waiting till 2 o'clock to get started. Today is a little bit different from last week. In that last week, if you remember last week, we had a limit of 300 students on the lesson, and so because we felt that we might well exceed that today, what we did was we decided we'd stream it through YouTube. So because of that, then if you're typing in questions into the chat, and we're delighted if you do, Declan won't be able to answer all of those live in the same way he would have last week. So what I'll be doing here once Declan takes over is I'll just be kind of keeping a note of the questions. A lot of them I'll ask Declan at the end of the hour, if they're general questions, and if they're specific, I'll send them over to Declan. I can communicate those to Declan. He'll be able to answer them at the time. So if you have asked a question during the lesson that hasn't got answered, that's probably because it's going to be answered at the very end. So with all that in mind, you can see here, so there's a couple of things. Oh yes, you can see, so our guys behind the scenes there, we got Conrad and Brian and Peter, who are here, are administrators. And you can see there they put up the link for the free business pre. So you just click on the SurveyMonkey link for that. So that's going to be on the 11th. So it's going to be the Thursday morning, 9:30 to 12:30 when business should be on. I know some of you are going to think, now what about the other subjects? Have we got stuff in line for that? We do. Partly, we're just making sure we have everything lined up before we announce all those little bits and pieces. And yes, exactly. If you could put in those of you who have the results for Maths pre-paper 2, if you can input those results today, that would be fantastic. Let me think what else I was going to say. Well, I suppose at this point, we're just coming up to 2 o'clock, and we're delighted today to have Dr. Declan Cathcart. Declan teaches in Temple Carrig School in Wicklow, and he also lectures in UCD, where a lot of you will be hoping to attend next year. A lot of you will recognize Dr. Declan Cathcart's name. You'll say, where have I seen that before? Declan's the author of Active Science and Biology Extra, so a lot of you here today will recognize the name through that. I know Declan does an awful lot of revision courses for students in the Leinster area, and he also does a lot of CPD for teachers. So for all the teachers who are tuning in today as well, you're very welcome. And you might well know Declan through those CPD courses for teachers. On that note, it's just coming up to 2 o'clock, and I think it's just reached 2 o'clock. And on that note, I'm going to hand you over to Declan, and I'm going to sit back here and learn a bit of biology myself.
Thanks very much, Owen. That was a very nice introduction. Can I just say I'm delighted to be here. Welcome to everybody who've taken the time out on a sunny Sunday afternoon. I'm looking outside here and the sky is blue in Wexford anyway. So yeah, it's a privilege to be here. And like I said, thank you for turning up to do some genetics on a Sunday afternoon. I'm hoping that it's going to be, it's only an hour's worth, and we'll be able to get through a good bit of material. Hopefully I'll be able to show you some tips and tricks on how to tackle this topic, which is a lot of people find challenging enough, I suppose. I think it's a great one. If you can answer the genetics question in an exam, it's one of those questions it's one of those topics that if you get your head around it, and you kind of practice the crosses and you understand the concepts, it's the kind of question that won't take you very long, and you can get full marks in a question. So it's worth spending the time on. It's one of those topics as well that, genetic crosses, genetics in general, I suppose, is one of those topics that often comes up on the section C and the big questions in the Leaving Cert exam. So it's worth investing the time and the effort, I think. So hopefully this will go some way towards helping you in that cause, right? So a lot of definitions. There's a lot of heavy-duty technical terms in this chapter as well, and same with biology, the whole course in general, right? Definitions are something that you should really, make a sort of a side project in your studies when it comes to your biology revision. And if you do a search just on that, if you do a search in Quizlet with my name, searching Google Quizlet Declan Cathcart, then you'll see there's a monster definitions Quizlet, which should be a complete set of all of the Leaving Cert definitions. It's worth, like I said, taking that as definitions as a little side project and do them in bite-sized amounts, 10 or 20 every few days or every couple of days over the coming weeks. It's well worth, again, the time that's invested. So there's a lot of definition in this chapter, and the first one obviously is that genetics is the study of inheritance, right? We are going to focus today particularly on the genetic crosses, so what's often referred to as the Mendelian genetics, because Mendel is the father of genetics. We'll talk about Gregor Mendel in a minute. I often find that when people are asked this, who's the father of genetics, they confuse Gregor Mendel with Charles Darwin. So they were obviously they never knew about each other's work, which was unlucky, because it would have made sense of their work if they'd known about the other one's work, but they never did, even though they were contemporaries and they were they were studying and they were publishing at the same time, but they weren't really available. Darwin wasn't available of what Mendel was doing. So Mendel was studying away in his garden in Hungary while Darwin was in England. Mendel didn't know about this, what I'm showing you on the screen here at all. So he wasn't aware of what we know now. And so it's much easier for us to make sense of what Mendel was doing than it was for Mendel to make sense of it, which makes his achievement much more remarkable. So we know, for example, that every of our body cells has 23 pairs of chromosomes. So our chromosomes come in pairs, and that those pairs, each of the gametes that are made receives only one of each of those pairs, right? So we know that now, and we know that before we even come to our study of Mendelian genetics. But just as a reminder, so like I said, the reason is that our gametes end up with only half of the number of chromosomes compared to our body cells, is that each of the gametes only receives one of a homologous pair. So each of the gametes, in the case of humans, receives 23 chromosomes, but they are just individual chromosomes from the pairs that exist. So, like I said, Mendel not aware of any of this. So we're going back to 1822, and as it turns, he tried to be a teacher, and he couldn't get in. They wouldn't let him in. He kept failing the exam, so he gave up on that after a couple of tries, and ended up becoming a monk. He must have been fairly good as a monk, because he became the abbot of the monastery after some time, and in the meantime, he became a very keen scientist, a very keen researcher. You know, he's one of the kind of, you know, like a good example, let's say, of what you might call of good scientific method or good scientific practice. So a lot of his discoveries, of course, weren't really, generally people weren't made aware of them until after he died. So he didn't get the kind of credit while he was alive for all of the amazing work that he did. So he chose peas as his subjects, and the reason he chose them is because they have a short life cycle, they're easy to grow, short generation time. You can grow a massive number of plants, which is what he did. Massive number of plants meant that he could replicate his experiments, right? So replication is a big thing. He grew them because they can be self-pollinated, and we'll see that that's a big feature in a lot of the experiments that he was able to self-pollinate plants that he'd self-pollinate offspring of crosses that he'd done. He started off with these, and he kept populations of these true breeding strains. So true breeding strains are strains that if you take a true breeding plant and you self-pollinate it, that you will always end up, all the offspring will be have the same characteristic, the same phenotype characteristic as the parent does, right? So these were important. He didn't just do one trait either. So he did, with the patience of a saint, you might say, he did all of these different traits. So the shape of the seeds, the color of the seeds, flower color, the position of the flowers, color of the pods, and the height of the pea plants. So as a scientist, just in terms of scientific method, and that's one of our things we must know about, he really sort of ticked a lot of the boxes. He was a very keen observer. He took very large number of plants, so replication was a big thing, so that he knew his data was reliable. He recorded everything meticulously. And then he looked for those patterns that would only be apparent because of the huge numbers of plants that he did. And then he was able to see the patterns and convert those into simple ratios. And it's that gave him the insight. So he took one of his early experiments was he took pea plants, and for example, with flower color, he took true breeding purples and true breeding white flower plants, and he crossed them, and he found that 100% of the offspring, and I'm sure you know this, this is just a quick run through, 100% of the offspring turned out to be purple. So that was a mystery in itself. But then when he went further and he took those F1 hybrids, the filial generation 1, took those hybrids and he crossed them with themselves, in other words, self-pollinated them. He took this F2 generation, and when he did the numbers, when he started counting the numbers, and he didn't just do this with flower color, he did this with all kinds of different characteristics, he kept coming up with this magic 3:1 ratio. And so he was trying to figure out, how is it that this happens, right? He didn't look at, like I said, just flower color. If you look at this chart here, he did it with, round and wrinkled seeds, yellow and green seeds, the position of the flowers, tall and small plants. you've seen these characteristics come up in past paper questions, right? So you should be aware of them. And in each case, it's the ratios. Look at these numbers of plants. He must have spent a lot of time in his garden anyway. That's for sure. So he figured out these ratios of 3:1. So he was puzzled by this, obviously, right? So the F2 generation worked out to be 3:1, and he had to try and explain why this was the case. So what he did was he came up with this. This is the first law, first of Mendel's laws, the law of segregation. So he said that it must be, the only explanation he could figure was that each characteristic is governed by a pair of factors, and that these factors separate during gamete formation so that each gamete receives only one of each of the pair of factors, and that these factors then obviously the pairs reestablish at fertilization. So the first law is just that these, and he called them factors, we now know they're genes, but that each gamete, that during gamete formation, each characteristic is governed by this pair of factors, and that they separate during gamete formation so that each gamete receives one of each pair. Okay, so that's sort of that law applies to Mendel's the monohybrid crosses, when he's just tracking one trait or one characteristic at a time. So the explanation now that we now know much more detail about is that this is how you can look at it, if you like, if you look under the hood of what's going on inside the cells of the plants, that the characteristic that is governed by a pair of factors, what we call alleles now, different versions of a gene or alternative forms of a gene, and that in the case of the purple tree-breeding plant, both of those alleles is identical. They're both of the alleles code for are purple genes, right? Whereas the small p is, and the reason we use big P is because that's the dominant trait. We'll come to dominance and recessive in a second. The small p is white, and what happens when you when these gametes, when these alleles are combined, then after in fertilization, is that one of the alleles is always dominant over the other. In this case, the purple is dominant over the white phenotype. So this was the explanation that Mendel offered, and of course he was right. So when it came to his second cross, when it came to crossing the F1s, self-crossing, self-pollinating, his explanation then his thinking offered a reason for why it was that in 25% of the cases, the white phenotype popped its head up again in the second filial generation. So some of the plants ended up, some of the offspring, some of the progeny ended up being white, and it seemed to be always, roughly speaking, 25% of the time. So 3:1 ratio. So these are the definitions. You need to know these definitions. So we talked about a gene. A gene is simply a section of DNA that encodes a specific characteristics, and that really just means that generally speaking, it codes for a particular protein. An allele is an alternative form of gene, so those factors that Mendel was talking about. Homozygous, that's those true breeding strains we talked about are homozygous strains. So in one case, in the case of the purple plant, it's homozygous dominant. So because the dominant gene, the dominant allele is an allele that's fully expressed, whether it's present in the homozygous or in the heterozygous condition. So if the purple gene, the big P gene, was present with either another big P gene, or if it was present with a small P gene, the recessive allele for white, didn't matter. That was what comes through in the physical appearance and the characteristics of the plant, right? Recessive, on the other hand, and be careful of these definitions, right? It's attempting to start using your own kind of understanding of it and talk about one masking the other and whatever. I think you're better off just sticking to these sort of textbook definitions. So whereas the dominant allele is fully expressed in either the homozygous or the heterozygous condition, the recessive allele is one that's expressed only in the homozygous condition. In other words, when it's present with another recessive allele, okay, and it's not expressed in the homozygous condition. So these alleles then are alternative forms of the genes, and what we call genotype is is generally speaking, it's the genetic makeup of an organism. But obviously, the genotype of an organism is not necessarily all of what's going to be responsible for its physical characteristics, right? And so what we call the phenotype. So the genotype is represented by, in the last cross we did, big P. That's a genotype of a plant, and the phenotype, the physical appearance, turned out to be purple. And generally speaking, genotypes, of course, affect phenotypes. But when it comes to phenotype, just be careful that you do understand that there's an environmental component here. So tall plants are tall if they have, the genes, if they're expressing the genes for being tall. But if they're not, if their environment is not where it's not getting enough sunlight, or they're not getting the right kind of nutrients from the soil, then they're not necessarily going to be that tall, right? So of course the environment the phenotype is a combination of genotype and the environment. Okay, so whereas the phenotype is tall, the genotype, in both of these cases, the genotype, you can't necessarily tell whether the genotype is going to be homozygous or, heterozygous or homozygous dominant. Just on that, on the heterozygous, there is no, you can't, it makes no sense to talk about, and just be careful with the language, to talk about heterozygous dominant or heterozygous recessive. There is no such thing. There's just heterozygous, whereas there is homozygous dominant and homozygous recessive, right? So just skip over it. This was Mendel's first law then, in language that we understand. It's just to put a bit of a sort of explanation behind it then. That we would now know that alleles of a gene come in pairs, and that they separate during gamete formation, and each gamete receives only one of each of the pair of alleles, and then that those pairs are formed again or made again when the gametes fuse during fertilization. Right? What again Mendel wasn't aware of is that the reason for that is because what we know, there are these, and I'm going to talk about at the beginning, these pairs of homologous chromosomes. So each of the pea plants was receiving one of each of these homologous chromosomes from each of the parents, right? From each of the parents. So when you look at chromosomes, there's a few things then that you can more definitions you can remind yourself of. So centromeres, if you go back to mitosis, this is the part of the region of the area of the part of the chromosome that attaches to the spindle fibers during mitosis when they get pulled apart. The locus of a gene is the physical position, that its location on a chromosome, right? So that's the locus of a gene. So plural of that are loci. So these A and T and R and B, these are different loci on chromosomes where these genes are positioned, right, or located. So in this case, for this pair of chromosomes, this is homozygous. Sorry, this is heterozygous for A. It's homozygous dominant for T, and it's homozygous recessive for R, whatever those genes are, and again heterozygous for B. Okay. So I'll skip over that. Mendel's second law. So Mendel didn't just stick with, of course, he started to make his life complicated by now tracking, studying two characteristics at the same time, and how they are inherited together, right? So this is what we call dihybrid crosses, where you're studying the inheritance of two different characteristics simultaneously. So dihybrid crosses are something that he did, and it's dihybrid crosses that led to his second law, which is the law of independent assortment. So I'll come back to that in a second. No, I won't come back to it. So what he was saying, what he realized, and I'll show you the sort of the evidence that he had for that. He realized that when gametes are formed, it's either of those pair of factors is equally likely to combine with either of another pair of factors, right? So those factors being alleles, right? So alleles are coming in pairs, and it's just as likely as one of those pairs of alleles is just as likely to combine with either one of another pair of alleles when gametes are being formed, okay? So if you look at one of the crosses that he did, these are round wrinkled peas, round and yellow peas, I should say, and there's two different genes or two different pairs of factors that are involved in the shape of them and in the color of these peas. So if he crossed these true breeding plants that were round and yellow, producing round and yellow seeds, and he crossed them with another set of true breeding plants which were producing wrinkled green peas. Then he found, similar to his earlier monohybrid crosses, he found that 100% of the time he got round and yellow. So he knew that round and yellow were the dominant characteristics. When he did the second cross, when he did this, took the F1 generation and he crossed, self-pollinated those plants, he ended up with this. This is the ratio that you need to just sort of, burned in your memory when you go into the exam, is this 9:3:3:1 ratio. He found that the phenotypes cropped up again and again in 9:3:3:1. And how he was able to deduce that from the numbers is it's just a testament to his genius, because how you'd be able to realize that this is from the huge numbers that he was doing, that these simple ratios of 9:3:3:1 were coming through again and again. He must have done a lot of experiments, right? So he found that the ratio of round and yellow and compared to round green and so on were 9:3:3:1. He also noticed that there were new combinations compared to the parents, and there were different, that these new combinations, in other words, that they were different phenotypes compared to the parents, right? So where you start off with round and yellow or green and wrinkled parents, now you were getting combinations of those. So you were able to get round and green and wrinkled yellow. So this was a new thing, and this is his explanation for it. So this is the cross done, and I'm going to show you this, what I would call the 8-step method that I would recommend that you when you're doing crosses, even no matter how simple the cross is, and most in your studies, but definitely in the exam situation, if you're going to tackle a cross and it's something like this, but to be honest with you, any cross that you need to do, crosses are generally treated like labeled diagrams. So I would advise, and I'm going to show you how to lay out crosses with this this method that I would always use for every cross. So that's just another version. One last thing, just on the genetic crosses that Mendel did. He, sorry, not that Mendel did, on dihybrid crosses. Sometimes it does make a lot of sense to imagine, rather than just writing your crosses out in with letter codes, right? But to draw these simple chromosome diagrams, little stick chromosome diagrams. It's a question that they can ask you to do, to actually draw the chromosome diagrams for these. So it does make sense to be able to be able to do it. It also can help you make sense of what's going on in a cross, especially when it comes to what we'll talk about in a little while, which is cases where genes are linked. In other words, where two different genes are present on the same chromosome. So it turns out that Mendel was pretty lucky, really, because he happened to pick all of the characteristics, at least this is from what we can tell anyway, the characteristics that he chose to study. In each case of his dihybrid crosses, he chose pairs of characteristics that happened to not be present on the same chromosome, right? They weren't what we call linked. So they were on different chromosomes, and so they always assorted independently in the way that we just talked about it, Mendel's second law. So that's not always the case, and we'll see that in a minute when it comes to linked. But this just shows you a little bit more kind of explanation about why it is that the genes, or that the alleles rather, segregate. So that if you cross now one of these round yellow F1 generation plants with one of the original parents, the homozygous recessive parent, that you get this now a combination, or you get this even 1:1:1 ratio of these four phenotypes. And the reason being is because it's just as likely, if we look at this gamete over here, it's just as likely as that this that this chromosome carrying the recessive little r gene, right, the gene for the allele for wrinkled, that's just as likely to combine with a big Y as it is with a small y, right? So when it comes to these combinations, it's just as likely that either of these two different chromosomes will combine with any of these other pairs of chromosomes during fertilization, right? Incomplete dominance then. I do want to show you one cross, and the cross that I've decided to show you, and I'm going to do it out in front of the document camera then in a second. It involves incomplete dominance, but it's a dihybrid cross. where it's a cross that involves incomplete dominance, and it also involves a dominant recessive characteristic. Couple of questions just coming through there. I see Owen has just alerted me to them. Does genetics come up often, and is it well answered? It's I'm going to come to something that is particularly poorly answered in a second, which is sex linkage. But is genetics come up often? Yes, is the answer. It does. Is it guaranteed to come up? No. It depends what you mean by genetics. So what I would call, if you look at genetics, apart from the sort of the crosses, you're also looking at all the DNA technology stuff. So you're looking at the genetic crosses, you're looking at DNA profiling, genetic engineering. You're also looking at the structure of DNA and DNA replication. But you're also, what I would call included in the study of genetics is protein synthesis. So that molecular biology, which is transcription and translation, and also then, of course, the related and the topic that makes sense of all this in the first place, which is variation and evolution. So if you talk about genetics as being an umbrella term for all of those, all I can say is it would be very likely that some of that is going to appear on a question, and it's likely in section C. It's often the case that genetic crosses might be part of a big question in section C, and it might be coupled with a question on evolution and variation, or a question on DNA profiling or structure of DNA or something along those lines, right? So genetics is one of those questions that, yeah, very likely is what I'd say. Okay, so incomplete dominance. If you take, in the case of some characteristics, if you take, for example, snapdragons, if you take red snapdragons and you cross them with white ones, you get pink progeny. So that's because of this phenomenon that we call incomplete dominance, right? And I would suggest to you that and it's, this is not something that you may not agree with this, and you might decide, look, I'm perfectly fine, and if you've done it a different way in class, then maybe that's a better way to stick with it. I like to use this rather than just the way of writing down alleles that we've been doing up until now for the dominant recessive situation. When it comes to incomplete dominance, just so students don't forget that they're dealing with an incomplete dominant situation, I would use this way of sort of coding those alleles, right? So for color, it's either red or little r. You could use W as well. I think that'd be just fine. And sometimes I would do either one or the other, and I might use W today just to show you what I'm talking about. But in the situation, in the case of incomplete dominance, what happens is when you have a pure breeding strain, a pure breeding homozygous red strain, in other words, if you self-pollinate that snapdragon, you'll always get red. And similarly with the white one, it's a pure breed. If you, it's a pure breed or true breeding, right? If you take it and you cross it with itself, then you'll always get white plants. But if you take one of each, then you get a whole load of pink plants. In fact, you get 100% pink, because the combination of big R and small r in an incomplete dominant situation gives you this this pink phenotype. Okay. So it's heterozygous for the genes, heterozygous genotype, but it's a pink phenotype. So this is what's going on. So here you can see, you can use the R for red and the W for white, and I think that's that. But I would avoid, personally, I would avoid using just big R and big W without the C. I think the C with a superscript of the R or the W or R or little r, for me that works better. I think it makes more sense, and it separates clearly in your mind an incomplete dominance question or problem or cross that you're doing out with a dominant recessive kind of one, right? So this is the way. This is again back to our 8-step method, and I'm going to show you across in a second, but just to go through some of these incomplete dominant situations. So these are the two purebreds, and 100% of the time, because there's only one possible kind of gamete that comes from either of these parents, when you combine these two, you get all pink, and this is the phenotype. Okay, so C big R, C little r, or CW. In this case, you take the two pinks and you cross them, and it's a similar sort of situation to where we were getting the 3:1 ratio, but the phenotype ratios now are different because we're dealing with an incomplete dominant situation. So the genotypes still come through the wash similarly. So you get 2 big R's, you get 2, sorry, so you get 1 homozygous red plant. You get 2 heterozygous pink ones, and then you get 1 homozygous white plant. Okay, so the ratio of phenotypes is 1:2:1. So it's a good idea to write this last stage of the ratios. You know, sometimes there is no ratio, it's just 100%, but it's a good idea to just write that there at the bottom anyway. So another version of that would be if you take one of the red ones and you cross it with one of the heterozygotes, and you will get you'll get 2 reds and 2 pinks. But that's the phenotypes. As it turns, the genotypes work out to be 2 homozygous dominant, homozygous red plants, and then you get 2 heterozygotes. They're the 2 pink ones. Okay. So I'm going to switch now to the other camera just to show you this. If you have the paper in front of you, I can put it up on the screen, but because we're on YouTube here, it's not easy for you. You aren't able to control the screen. So what I'm going to do is I'm going to switch cameras, but I'm going to share with you. This question is from 2011, and it's question 13 from 2011. So it's going back a bit. And as far as I can tell, they haven't asked this kind of a combination of dominant recessive with incomplete dominance in a dihybrid cross before or since. You can correct me if I'm wrong, anyone out there, but I think this is a good one to show as an example for this 8-step method that I'm talking about. So let me just switch the cameras here, and hopefully you'll be able to see that easily enough. I will just focus on the page there. And now what I'm going to do is I'm just going to stop sharing. I'm going to do a new share with the question that I'm talking about. So let me just get rid of that. And so hopefully you're able to see this question. This was, like I said, a question from 2011. So if that's on your main screen at the moment, hopefully it is, just to look through it. So snapdragons again, and there's no dominance between the allele for red and the allele for white. So that's just what we were talking about. That's the incomplete dominant situation. So the heterozygous individuals have pink flowers. The allele for tall is dominant to the allele for short, just as it is in peas, and we've been seeing that. And these pairs of alleles are located on different chromosome pairs. So that means, in other words, that they're not linked, and we'll talk about linkage then in a few minutes. So I'm going to skip that and come back to that. The significance of the fact that the 2 allele pairs are located on different chromosomes. No, I won't skip it. We'll talk about it more in detail later. The significance of it is that they don't, that they segregate during gamete formation. In other words, that they obey and follow Mendel's law of independent assortment, his second law. Right, so that's the significance of the fact that the allele pairs are located on different chromosomes. If they weren't located on different chromosomes, then they wouldn't segregate independently of one another. So here's the cross. So it's a plant which has pink flowers. So we know from reading the question it's heterozygous if it is pink, and it's heterozygous in terms of stem height. So we should be able to figure out then, because we know it's heterozygous, we should be able to figure out what the phenotype, what the appearance of that plant is. Okay, it was that those are the 2 parents, and it was crossed. A plant which is that's one of the parents, and that was crossed with another parent which has white flowers and a short stem. So we need to get that going. I'm just going to switch then. If you can, hopefully you're seeing my document camera here, and I'm going to just bring you through this 8-step method. So I did say to you already that when it comes to the biology exam, that there's usually a lot of marks going for doing out one of these crosses. It's treated like a label diagram, so there are different parts of it. Often, that you need to, that I would always say that it's a really good idea to put these headings, if you like, to each of the steps. And so often what happens is, if you do the cross out, you end up answering all of the questions that are asked after that, right? So once they've explained what the cross is in the question, and if you go and then you do out the cross fully in this way that I'm going to show you, that you have answered most of the questions, if not all of them, that follow on from that. Okay, so in this case, what we'll do is we'll ignore the questions for now. We'll just do out the cross, and then we'll see what happens. So the first of these steps that I would talk about is to write down the parent phenotypes. The parent phenotypes in this case, we were told, it's tall, round, and the other phenotype is small and wrinkled. Okay, so it's tall, round, small, wrinkled, and we know again from the question. Sorry, I apologize. I'm looking at the wrong thing here completely. Where are we? Start that again, shall we? So we have, first one is parent phenotype, and we're looking at these snapdragons, and the first parent we're told is tall and pink, and the second parent is short and white. So when we look at parent genotypes then, and like I said, it's a good idea to really label these out. The parent genotype for this one, if it's tall and pink, then it must be, let's look at the tall first of all. We're told that it's a heterozygous, it's heterozygous in terms of stem height. So it must be big T, small t. And it's pink, so there's no other way around it other than that it's C big R and C little r. If you want, you could use a W here. That'd be totally fine, and maybe prefer to do that. So the short white one, it can only be short because it's a dominant recessive situation. It can only be short if it has two small t alleles, right? It can only be white if it has both of these, the alleles are for white as well. So if you then go through the stage of meiosis, in other words, the generation of gametes from these, there are going to be 4 different possibilities for gametes coming from this parent. Okay, so you will write those 4 down. This then will give us the gametes are produced then by meiosis, right? There's only going to be one possible kind of gamete from this this parent here. So let's just sort that one out first of all. That's going to have to be, it can only have a small t allele, it can only have a white allele for color. We put circles around our gametes, just good practice, okay? And examiners like to see that as well. So the 4 gametes that can come from this, remember, this is Mendel's second law, that during gamete formation, either of a pair of alleles is equally likely to combine with either of another pair of alleles, right? So that big T is just as likely to combine with this allele as it is this factor as it is with this factor. So we'll do that. We'll put in all the different combinations. You can have big T with big R. You can have big T with small r. You can have small t with big R. And you can have small t with small r. So there are 4 gametes from this parent and only 1 from this parent. What we do then is, when we're doing our, and you know this hopefully at this stage, you're doing all about the fertilization. Let's get the spelling right. You're all about the Punnett squares, right? So fertilization is a vehicle. It's a method for being able to show all the different possible combinations of zygotes that can result from the fusion of these gametes. Okay, so the Punnett square in this case, though, is going to be fairly limited because we're going to have 4 different possible gametes sitting up here. We'll have, rack these 4 up here. We'll have T, this one here. I'll just list them out quickly. You're going to cross those with each of the with these here, with this one gamete from the other parent here. So we'll put our circles around those, and we'll put in the offspring that results then into our square. So in here goes combination of these, and I'm sure you've done this often enough to know what's going to happen now. So all of these factors then are combined during fertilization. Okay, if you can read my scrawly writing there. So the next stage is to write down the genotypes of these progeny. Okay, so the genotypes of the progeny are basically what you've just written into the boxes. Now, it seems like, why would you bother? Just after, you know, writing them all here, why would you have to write them all out again? And the reason I would say that is because you've just done a fertilization step, but you need to say what these all are. And these are the genotypes of the progeny. So basically, it's a labeling exercise. And like I said, treat this as if it's a labeled diagram. So write down it also means that if you, like I did, if you are a bit, you know, if you've had to squeeze them into your box because there's a lot of lettering going on here, then you can write them up more clearly. So the second one is, third one is, got them the other way around, that's all right. And then the last one is this one here. So these are the 4 different genotypes of the progeny that you pull out of your box. You then explain what the phenotypes of those are, the corresponding phenotypes of each of these. And in this one, you can see this one is going to be tall and it's going to be pink. So tall, pink. This one here has to be small, but it's also pink, so short, let's say. Pink or dwarf pink. This one here is going to be tall and it's going to be white. And this one here is going to be short. Oh. It's going to be short.
Declan, sorry to interrupt there. Just one thing, that the screen that we can see was with the main screen that we were seeing was the text.
I just realized, yeah.
Oh man, no, perfect. Oh yeah, I can see it. Thanks,
okay.
Declan.
So that's unfortunate now because the I'll go through it though. So let me make sure. Yeah. Okay. So let me just finish this off and then I'll bring you through it. So short and white. Okay, so just to go back through this then, and that's unfortunate that you weren't able to, you know, working on Zoom, that you weren't able to see through YouTube that that the screen for you was small. But having a look through this then, so I'll just run through it again. So the parent phenotype was tall pink and short white. That means that the genotype was, and we can tell from the question that they told us it was a heterozygous for tall. So it was tall, and we know because it's pink and it's an incomplete dominant situation that must be big R, small r. The short plant can only be 2 small t's, and because it was white, we know that this is going to be it's the homozygous situation for the color as well. So we then went through meiosis. The meiosis stage is the generation of these gametes. In this case, and what I was saying was that because the genes aren't linked, because the genes aren't present in the same chromosome, that Mendel's second law applies, the independent assortment. And what that's saying is that it's just as likely for big T, it's just as likely for this factor here to combine with big R as it is with small r. Okay, and again, it's just as likely that the small t is just as likely to combine with either of those pair of factors. So that's Mendel's second law. This is Mendel's second law in operation here. So all of these 4 gametes are produced, and only one possible gamete can come from this parent here. When we do the Punnett square, this is the fertilization stage. So stage 5 is when you do you go through the possible outcomes of fertilization, where this gamete, the only one that's possible from one of the parents, is fused with any of those other 4 gametes that were produced from the tall pink strain. So you get these 4 different possible genotypes of the progeny, which we then list out, and I think it's good practice to list those out. the phenotypes then of the progeny was where we were just at, and you list those carefully, and you see that there are 4 different phenotypes. And you also notice that there are a couple of phenotypes that are new combinations of characteristics that are different to the original parents which were here. Okay, so these are new combinations. These are parental types. Okay, these 2 here. So 8, as I said, just to round things off, I would always just say is if there's a ratio, stick it in there. So just again, one more time, I would recommend that, and I've provided the notes that I've given you. You'll see that the crosses that I put in those notes are all done out with this 8-step method that I'd recommend you to do. Okay. All right. So where are we now? We're just. I'm going to switch back to the screen again, and we'll have a look at it. 2 more things. One of which is linkage, and the other one is sex linkage. Somebody asked earlier on that is it one of those questions that's well answered, and I'm just gonna stop sharing or stop this video here. I'm gonna just go back to my other video. Okay, so yeah, there's a report comes out from the chief examiner of the chief examiners in the SEC for biology, and a few years ago he put in a note of questions that were particularly poorly answered, and one of those questions, if you were with me last week, then I pointed out to you, was sex linkage, and it's just poorly understood, I think, is the main thing. So that's a different thing to what we're going to just look at now very quickly, which is sex determination. Okay, so I'm just going to start from here again and flick through this with you. So what we're talking about here is the fact that of our chromosomes, 22 pairs are what you call autosomes, right? We've got 22 pairs of autosomes because one of the pairs we refer to as sex chromosomes, and those sex chromosomes is the ones we're going to talk about. When you look at the set of chromosomes in a male, this is generally speaking, that males in humans have an X and a Y. You notice that the Y chromosome is worryingly small, lads. That's something that is getting smaller over time as well. So it seems to be evolving its way out of existence in humans, which is not fully understood. So in women, generally speaking, females in humans have 2 X chromosomes, and it explains then. And by the way, it's not always the case in other species. So in butterflies, for example, it's the other way around, where the XY combination is what determines the female, and 2 X's is a male in butterflies, and some other species too. So generally speaking, if the Y chromosome is present, you get a male in humans, and if it's absent, then the person is female. Okay. So when the X and Y chromosomes separate, then in gamete formation, half of the sperm from a man end up having an X chromosome, the other half have a Y chromosome. Okay. Let's skip through this. So when you do the cross out and you focus just on the sex chromosomes, it explains then why it is that, generally speaking, around the world, we've got more or less 50% men and women are born, and it's because of this simple Punnett square, right? So the gametes, the eggs from a woman, both of which contain the X chromosome, and gametes from a male, 50% of them contain an X and 50% contain a Y. So when you come to the fertilization situation, you get 50% female and 50% male. So that's the explanation for why that is. It's a favorite question. Okay. So coming to linkage, then, this was one of the things we wanted to talk about, and one of the last 2 things we'll talk about today, anyway. So linkage is the situation where genes for different characteristics are physically located on the same chromosome. If genes are present, two different genes for different characteristics are present on the same chromosome, they are said to be linked, right? So I said to you earlier on that they don't assort independently, and this is the reason why. So in the linked situation, if you had a situation where two different characteristics are coded for by genes, and those genes happen to be present on the same chromosome, then this is one pair of homologous chromosomes. Okay, so in this case, the dominant alleles for both A and B are on one of the homologous chromosomes, and the dominant, the recessive versions, the recessive alleles of those are on the other of the pair. So when it comes to gamete formation, there are only two possible gametes can be formed. So big A and big B are, they're attached to each other. They're stuck with each other. Okay, when it comes to gamete formation, and the point of it, this is they don't assort independently. Okay, so they don't fit Mendel's second law of independent assortment. So in the non-linked situation that we've looked at already, you can see that the outcome is completely different when it comes to gamete formation. Mendel's second law, what he always found with all his crosses, was that it's just as likely for big A to combine with big B as it is big A with small b. In other words, those alleles, either of those alleles is equally likely to combine with either of those other alleles here, right? And that's Mendel's second law. So you get 4 different possible gametes in the non-linked situation, and that's the situation we've been looking at up until now with these dihybrid crosses we've been doing. So you see that the linked situation limits the number of combinations of characteristics, combinations of genotypes and phenotypes that you can get when breeding, right? So that's what that, and it's a favorite kind of question in a cross is, either one of two things: either explain what would happen if the genes were linked, or they've given you maybe there are questions where they will give you a cross, and they tell you what the outcome of this cross is, and when you do the cross out, a dihybrid cross I'm talking about, if you do the cross out, you don't get and you know how to do it, and you do the cross out, you're not getting the ratio that they're saying was the result of the cross. Okay, and the reason for that is because the genes are linked. All right, so they're the kind of two questions that you often see. So sex linkage, this is the one that was really badly answered in the view of the chief examiner, and obviously that's just based on the evidence of how the questions were answered on sex linkage over the years. So this is not really to do with linkage at all. This is to do with the situation where we were talking about X and Y chromosomes, and if a characteristic is controlled by a gene that's on a sex chromosome, either an X or a Y chromosome, we call it sex linked. Now, because the Y chromosome has so few genes on it, generally speaking, what you're talking about is that it's controlled by a gene on the X chromosome. Okay, so they're sex linked. Genes on the X chromosome are called sex linked. When we look all the sex linked genes that we look at are ones that are present on the X chromosome. Okay, so if a gene is sex linked, it means that men, because they only have one copy of the X chromosome, they're only going to only have one copy of that gene in their genome, right, in their cells, unlike all the other genes that they would have, which they would have 2 copies of. So this is why it is that so-called sex-linked conditions, conditions, medical conditions that are associated with genes that are sex-linked, that are present in the X chromosome, they affect men much more than they affect women, because if it's a dominant recessive characteristic, then women can be carriers for it and not have any issues. Whereas if men receive that one copy of that recessive gene and on their X chromosome, then they're going to get the condition. So let's look at that just quickly. Most of these mutations are recessive. The 2 classic ones, and you need to know 2, are hemophilia and color blindness. So I've done crosses on color blindness, and they should be in the notes. So color blindness is to do with the red, green, blue receptors in your retina, and it is an X-linked a sex-linked condition. Okay, so skip on from that. Just to look at the cross then, I think is the best thing to do. So again, we're looking at this 8-step method. So when you're looking at sex-linked crosses, again, you're looking at X and Y chromosomes and tracking those, and you're using a superscript of a big N for the normal vision and a small n for the colorblind allele, or the colorblind mutation, the mutation that causes colorblindness, all right, that allele. And when you do the crosses, then if you were say to a male with normal vision and a female with normal vision, but happens to be carrying one copy of the recessive gene for colorblindness, then if you do that cross out, what you end up with is you do end up with 25% of the time you're going to end up with a colorblind child. So the ratio is 1:2:1. 2 females, neither of whom are colorblind, and 2 males, it's a 50-50 that one of those male children will be colorblind. Okay, and then there's a few other crosses, a few other versions of that. It's definitely worth your while doing out these crosses, starting with different combinations of phenotypes of parents and seeing what you can figure out from doing the cross yourself, and then go back and check with the crosses. So there's only so many different combinations of these crosses for sex-linked conditions that they can ask you. So if you do these few of them, you should be covered, right? The other one is cystic fibrosis, right? And that's a particularly interesting one in the Irish context because it is much more common. It's a rare condition, but it's much more common in Ireland than it is around the world. Okay, it just seems to be much more present in our population. But the sex-linked condition of haemophilia and colorblindness, like I said, they're the two that would be good to know. Last thing I'd say to you then is about non-nuclear inheritance. So don't forget that not all of your DNA is inside the nucleus in your chromosomes. You have this extra chromosomal DNA. So and it's important to notice here because for humans, the mitochondria that we inherit are only, and the mitochondrial DNA, right? There's DNA present in the mitochondria that encodes enzymes that run for respiration and whatever. But that small amount of DNA that's present in your mitochondria is present in the egg during fertilization. Okay, it's present in the female gamete. There are mitochondria from the female present in the egg. When the sperm fertilizes the egg, only the nucleus of the sperm in the head enters the egg and fuses. That nucleus fuses with the female nucleus. The mitochondria, which are present in the midpiece of the sperm, don't figure then. They're left behind, right? So it means that there's in our history, in our DNA history, there's this continuous maternal line where the DNA in everyone's mitochondria only comes from the maternal line, only comes from your mother and her mother and so on. Okay, so this is a maternal line of, and it's been very important in trying to track, the history of the evolution of humans, right? In looking at maternal inheritance and mitochondrial DNA. But the same is true for chloroplasts in plants. They also have their extra chromosomal piece of DNA. It's present in the chloroplast, and it encodes photosynthesis enzymes and factors relating to photosynthesis. And it is also what you'd call, like I said, extra chromosomal DNA. Okay, we're going to call it a day there on the genetics. Other than I'd like to just, if Owen has any questions there that you've been firing at YouTube, I'd be happy to answer them. And Owen might want to have another word about the tuition center. I'm going to be running some intensive 3-hour courses once a week on different areas of the biology course, hopefully over the weeks then covering all of the course. So I'm broken it up into, I think, 5 or 6 3-hour courses. So hopefully 3 hours is not too long. I've done some full day revision courses there recently, which are fairly hectic, as you can imagine. And obviously you can't get everything done even in a 6-hour course, but in a series of 3-hour classes, and you could pick and choose whatever kind of topics than you wanted in combinations of topics within those classes, then hopefully that'll be something that I'll see some of you at anyway in the future. So, Owen, have you got anything for me there?
Sure, Declan. Firstly, let me say thank you very much. I think applied maths was across from biology back when I did the Leaving Cert, and I was more of a mathematician than a scientist. So I went that way. So I learned an awful lot today. So that was great. So if I'm sitting here for the hour very interested, I'm sure the Leaving Cert students must be the same.
Good to hear. Good to hear.
Let me just ask one thing, and you can tell me if you already covered this, right? This shows a little bit of lack of knowledge here, but there was a question just saying, can you explain the chromosomal theory of inheritance? But I think you might have gone through that, but I'm not 100% sure.
Yeah, I'm not quite sure what that phrase means, but if I think if it's it is what we talked about. It's what Mendel sort of deduced from all the crosses that he did, and we've kind of uncovered that. Then that's a historical sort of a term, then I suppose is what you call it, and that's what we covered today. Absolutely.
Yeah, that's great. That's what I thought. I thought you had covered that, but I just wanted to make sure on that. Yeah. And a number of students are saying, we know that the notes are available here for the students, but a number of students were wondering about the PowerPoint. Could they get that? I know we sent out last week's PowerPoint for them, so I suggested them if they haven't filled in the survey yet, if they fill that in now, that they'll get that sent
no problem at all. Yeah, I'm happy to share that as well. Sure.
That's brilliant. So let me just think. No, okay, so we know there's a couple of questions have been sent to me about the tuition center in general. I'll deal with those in a minute, but there's a question here just as came in. Any predictions for the type of cross that may come up this year?
No, anyone's bet. No idea. There's no such thing as trying to look at patterns and whatever. Yeah, as far as what kind of cross, there's nothing much I can help you with. The only thing I would say is that cross there, that one we did where it was a combination of dominant and recessive and an incomplete dominance in a dihybrid cross, that hasn't come up since 2011. It's a long time ago. So but I wouldn't say any more than that, I suppose.
Yes. Very good. Well, look, just to say, I can see it here on my various screens. There's the feedback here is excellent. There's someone just commented excellent lesson there, and we can see it coming through on the Instagram as well. A number of students and teachers, let me say, sharing the fact that this lesson has been on live here today and recommending it. So Declan, thank you very much for that. I just see one question coming in from Abraham. I think as a teacher I could probably answer this one, but I'm gonna give it to you as a subject specialist.
Yeah.
Is it best to study everything to achieve a good result? Or in some subjects I know you can kind of fine tune the course a little bit, but in biology, is it best just to try and cover everything?
It is, yeah, it is. I mean, and it might seem like it's a huge course. Hopefully at this stage, what seemed like this big huge mountain of material that you spent 2 years covering, that actually it's becoming much more manageable, and it seems to have shrunk in size. And now that you're kind of have entered into a revision phase and everything. The only piece of advice I'd give you, Abraham, and anyone who wasn't at last week's free lesson, is that I would say it is very much worth your while becoming an expert in units 1, units 2, and the mandatory practicals. And the reason for that is just the way that the paper is structured. That there is, you have, of course, you've got choice on the paper, but the way that the SEC have to set the paper up, it must be the case that approximately 44%, so less than half of I'll say this the other way around. For about 80% of the questions on the paper have to be from Unit 1, Unit 2, and the mandatory practical experiments. So that's an awful lot of questions. That's 80% of the questions that you could do, right? When you come through the choices, it's possible for you, to do Unit 1, Unit 2, and the mandatory practicals, which is just under like half of the course, right? And you can get about 80% of what you need in terms of, you know, the marks. It's about 320 marks out of 400 you can get from just looking at those 3 things. Now, that's not saying much, because Unit 1, Unit 2, that's a lot of material. It's also very dense material, but it isn't the huge big volume that say human systems and for example is. So rather than taking shortcuts, what I'd say to people is, if you make sure that you're an expert in Unit 1, Unit 2, and the mandatory practicals, then it will give you a little bit of more choice maybe when it comes to answering the plant and the human system questions, because there has to be 3 questions in section C have to be from Unit 3, right? So in section C in the paper, 3 of those questions out of the 6 that they give you have to be from Unit 3. So Unit 3 is the organism biology, right? It's the plants, the humans, and the microbiology, the protists and the fungi and the bacteria. So knowing that that's the way the paper has to be set up, I think it does point to the, like I was saying, just make sure that you are really good at your, you know your Unit 1 and Unit 2 and the mandatory practicals like the back of your hand before you go into the exam.
I'll just ask one last question, because I know Declan has something else on as well today. So there's a question here about, it says, is the FOIL method good to use with your 8-step method, first, outer, inner, last? Is that relevant to it? No, it's actually a maths term, the FOIL method. So yeah, it's something I recognize from mathematics, but I don't think, it doesn't sound to me like it ties in.
Not that I know of. Oh, yeah. You're the maths expert.
Yeah, exactly. I recognize it from maths, but I still said, just in case there was a connection there. So for the student who asked that, I think we can say, no, those 2 things are separate. Declan, I'll just say, we can see the thank yous are just are flowing in there for the lesson. So, but I would say on behalf of the Tuition Centre as well, I'd say thank you very much. Declan mentioned that he's going to be doing revision courses for the next five weeks, and those are available to purchase on the website, so thetuitioncentre.ie. And we will, in a moment, there's just a couple of more general questions there, and I know a couple of people have emailed me with general questions there in the last hour or so. I can talk to students through that, Declan.
Brilliant. Listen, I'll just say thank you very much to Owen and to everyone who came along today. Hopefully now you can get out and get a bit of fresh air, and that nice, if it's still sunny, if it's sunny where you are, I'm going to try and do the same. And thank you very much, everyone. And it's been a pleasure, and hope to see some of you soon. Thanks, Owen.
Declan, thanks again. Thank you.